Everyone, welcome to my channel, and this video is about the mechanism of the acid base regulation. So what is the acid? The donator of the H plus concentration in the solution refers to as the acid, while the acceptor of the H plus ion, that is hydrogen ion concentration in solution, refers to as the basis. So three primary systems regulate the hydrogen ion concentration in the body fluids to prevent acidosis or the alkalosis. The acid base body fluid buffer, second one is the respiratory mechanism of the acid base balance, and the third one is the renal tubular or the kidney regulation, and we will see one by one. So first of all, acid base body fluid buffer. So buffer are the solution which ties up the extra H plus ion concentration in the present of the solution until balance can be reestablished, and it contains a bicarbonate buffer, phosphate buffer, and third one is the protein buffer. So the reactions are as CO2 plus water in the presence of carbonic anhydrase enzymes. So it forms the H2CO3, and which dissociates into H plus SCO3 minus, that is hydrogen bicarbonate and the carbonate ion. Plus HCO3 minus plus sodium ion forms the NaSCO3, sodium bicarbonate. So phosphate buffer is the main element of the phosphate buffers are the H2PO4 minus and the HPO4 minus. So protein buffer in the RBC of the hemoglobin Hb is an important buffer, and H plus HHB, that is hydroxyhemoglobin. So respiratory mechanism of the acid base balance, the increase in the ventilation eliminates CO2 from the extracellular fluid, which by mass action reduces the H plus concentration and increases the pH, called respiratory alkalosis, and if it is reversed, it is known as the respiratory acidosis. So third one is the renal tubule or the kidney regulation, as we have seen previous in the excretion part of the uh, body. So it is a third line of the defense and acid base rebalance. The pH of the initial glomerular filtrate is approx 7.4, that is GFR, glomerular filtration rate. GFR is approx 7.4, whereas the average urinary pH is about 6.0 due to the excretion of the non-volatile acid produced by the metabolic processes. So renal conservations by the alkali reserves SCO3 minus and the excretion of the acid, and it includes four mechanisms. First one is the exchange of the H plus from the for sodium of tubular fluid, and reabsorption of the H2CO3 from the tubular fluid, formation of the ammonia, and excretion of the ammonium ion in the urine, excretion of H plus as H2PO4 minus in urine. So we can better understand through this chart in easy way. Exchange of H plus for sodium of tubular fluid, and the reabsorption of the bicarbonate from the tubular fluid. In this figure, the glutamates enter into the renal tubular cells, which through a process of deamination and the glutaminase and the glutamate enzyme, the NH3, it goes to the tubular lumen, and again it associates with the H plus to form NH4 plus, and with the help of the chlorine ion, NH4Cl is formed and excreted in the urine. So formation of the ammonia and excretion of the ammonium ion in the urine. So this picture is about the excretion of H plus as the H2PO4 minus in urine. So same as the renal tubular cells, tubular lumen, and the blood. So all about this acid base regulation of the animals. Keep watching. Thank you.